Yesterday when I was teaching, I said that our body has got several genes, and each cell has got all 46 chromosomes. So all the 46 chromosomes will have all the possible genes which are there in our body. Initially it was thought that we have got somewhere about 80,000, 70,000, some said 1,40,000 genes. Because when it was studied that the E. coli, like unicellular bacteria, has got somewhere about 4,000 genes, then we are much complex than the E. coli. So it is believed that we have got much large number of genes, and it was expected, it was calculated by, or some guess was made by some of the scientists. They said some said 40,000, some said 70,000, some said 80,000, some said 1,40,000 genes are there. Whatever the gene, each cell has got all 46 chromosomes, so all the whatever number of genes are there, all genes are present in every cell. Now the question is that why this cell is behaving only like skin cell, why the other properties, or how the other properties are remain unexpressed. It can also be capable of making hydrochloric acid or trypsin or pepsin or lipase or saliva or tear or sperms or ova or whatever type of properties, because the chromosomes are there, and the chromosomes are there, that means genes are also there, and the genes are there, that means the capability is there. So the biggest question is how a gene is regulated. What tells that what gene has to be become activated and what genes not to be activated? That was a very big question mark. Because just merely studying about the chromosomes or the gene or the chemical nature of the gene or the location of the gene, that doesn't mean everything. Suppose you go to a bank. In a bank you find that there are several different employees are there. Some is sitting on the may I help you desk, some are on the cash teller, some are to receive the money, some are there to make checks, some are making drafts, some are making something, some are opening account. There are number of people. Now each people is designated a particular task for that particular day. Next day he may be in rotation. So the one who was sitting to make draft on the day one, he may be sitting on the teller in the second day, he may be sitting on the may I help you desk on the third day, he may be in the some government deposits on the fourth day. So that means the same person, but that particular day is carrying out only one function. Now the question is that is how so many genes which are found in our body, only a certain gene become activated and other genes are not activated. Moreover, the genes are not all the time active. Not all the genes are all the time active. Say for example, when a child is there, a boy of 1 year, 2 years, 3 years, 10 years, 12 years, 13 years, he don't have any sign of mustaches and beard. He do not have any sign of hair on the chest and the under armpit. Then slowly these things become active. So now the question is that when these genes were there, already there, and they had to function, what stopped it till the age of 12, 13, 14, 15 years? And now after these years they become active. Similarly, in our body, the hydrochloric acid is secreted in the stomach when the food reaches there. Now how it is? So there are so many questions were there. And all these questions were based only on one thing, that is how a gene's activity is regulated. The regulation of genes are taking place in the 4 steps. The regulation of genes are taking place in the 4 steps. That is, the first is at the transcriptional level, that is which particular segment of the DNA will transcribe into RNA and when. Second is the processing, that is the hnRNA which is produced, which may have some wanted and some unwanted segments, exons and introns. The introns has to be removed and the hnRNA has to change into a matured RNA. And the third is, that is the synthesis of protein, that is translation. And the fourth step is, that is the function of a particular, third number is, that is the transport of the messenger RNA which is there into the cytoplasm, and the fourth is translation. So at the 4 levels. Now we are not going to study all of them because it is the topic of entire genetics which you are going to study in the MSc level. Then Only these things you will be studying if you are having genetics as a major subject. So this is a very vast scope and it has completed in number of years. We are not going to study all that. The example which we are taking to understand the regulation of gene is called Lac Operon. Lac stands for lactose. And lactose is a disaccharide made up of glucose and galactose, and it is found in milk sugar. Lactose is a sugar found in the milk, disaccharide it is, and it is made up of glucose and galactose. So its digestion requires some enzymes, and the major enzyme which is there, which is required to break the lactose into glucose and galactose is beta-galactosidase. Okay. Now the question is, suppose this is a bacteria, E. coli. Now E. coli, as long as it is not come across a milk, it will not have this enzyme, that is the beta-galactosidase produced. That means this gene will not be activated. No sooner it gets surrounded by milk, it will start making that. So that means normally the switch was off, and when it come across a particular situation, it could make the switch on. This switching off and switching on capacity is called regulation. And that was first time understood by Francis Jacob and Manod. The Jacob was a geneticist, and Jacques Manod. Francis Jacob was a geneticist, and Jacques Manod was a biochemist. Together they performed number of observations and they come to know the Lac Operon. A Lac Operon has the following thing. It has, you all know that the bacteria has got only one chromosome. So in the one chromosome itself, it should have all the regulatory genes present in it, all the genes present in it. There is a P site here. There is an I site here, there is another P here, there is an O here, this is Z, this is Y and A. I explain their function one by one. This E, I stand for inhibition. Inhibitory gene. So I will write it inhibitory gene. Wherever P are there, they are the RNA polymerase synthesizing gene. RNA polymerase synthesizing gene. So here RNA polymerase and here RNA. The O is the operator, which will operate the functioning of these genes. Together these three, they are functional, but their function will not go outside. They are the back, what is called, if you go to any office, you will find there is a front desk who are dealing with all the problems. And there are some people who are working behind. The front desk people are just meeting the people, taking their problems and passing it to behind and getting their solution and passing. So these are the front desk who will be showing the action, and these are the regulators which remain behind. These are called regulatory genes. Regulatory genes. All of these are taking some sort of RNA, they will be called genes. So they are making RNA polymerase gene. This is making inhibitory gene which will making a repressor. This is again RNA polymerase, and this is an operator. So all these collectively known as regulatory genes. Okay. Inhibitory gene which will synthesize a particular type of RNA, and for that RNA polymerase will be made here. Operator requires certain type of processing, that for that the regulatory gene the repressor will come and bind here, and it will, so it is in fact a switch. This operator is actually a switch. When it is turned on or when it is turned off. When it is turned on, the process will begin. When it is switched off, the process will stop. So these are all these. This particular segment is called regulatory gene, and this particular segment is known as structural gene. S-T-R-U-C-T, structural. Structural genes will make messenger RNA. Now again, here it is a very important thing. Only a single messenger RNA will be made which will have information for 3 different proteins. A single messenger RNA having information for 3 different polypeptides. Such messenger RNA, such mRNA which have information for more than one polypeptide are called Polycistronic mRNA or polycystone, commonly it is known as. If one messenger RNA codes for only one polypeptide, it is called monocystone. If it can code for more than one, then it is called polycystone. So some new things which we have understood here, that is the regulatory gene, the structural gene, polysystonic mRNA, operator, and this is inhibitory gene. Now what are the function of Z? Z makes beta galactosidase. This makes an enzyme beta galactosidase. This makes an enzyme that is called permease. Y makes an enzyme called permease. ASC wherever is attached, you know that they are standing for enzymes. Okay, and the last one is transacetylase. So 3 genes, ZYA, 3 genes, they will make one messenger RNA which will have information for 3 polypeptides. Such messenger RNA which have more than one information for polypeptide are known as polysystonic messenger RNA, and here that Z, Y and A are standing for Z for beta galactosidase, Y for permease, A for transacetylase. Okay, the galactosidase will break the lactose into glucose and galactose, but it will break only when these molecules, that is the lactose molecule, enter in the cell. The entry of these into the cell is regulated by permease. The permease increases the permeability of lactose so that more and more lactose can enter into the cell. So it is increasing the permeability. And transacetylase, as you know, trans means changing. Acetyl is a group, so it is an enzyme which is responsible for transfer of the acetyl group. Now where it is required, that is not very much sure, it is not given in the book. But these 3 enzymes are made by one single polysystonic gene mRNA, one polysystonic mRNA. Genes are of course different. So these genes which are actually carry out a function are called structural genes, and these genes which will regulate the making or non making of these things, that is regulating their function, are called regulatory genes. Okay, and altogether this is called an operon, and this operon is controlling lactose, so it is called Lac Operon. And who gave the idea of Lac Operon was Francis Jacob and Jacques Monod. One more question, Jacques Monod. What is operon? A unit consisting of regulatory and structural genes. What is Polysystonic Messenger Rna? I mean more than one. That is polypeptide synthesing information. What is operator? That is the switch which will turn this process on or off. What is IR? This is inhibitor. Now I can stand for inhibitor or inducer both. So you have to be very careful. Here it is inhibitor. And what are P standing for? They are the RNA polymerase making engine genes. Okay, so this is what the process, the Lac Operon is. Now we shall study how it functions. How it functions. So I am just rubbing it out. Now I will not write again and again what stands for what. Okay. Now E. coli was there. Now E. coli are present in the form of spore in the air. You all have come across a situation where your milk must have spoiled. Why milk gets spoiled? The basic reason is that Lac Operon falls in it and lactose bacteria falls in it, and lactose bacteria will slowly convert the milk into curd because it makes lactic acid. Okay, so that is their function. Now we are not studying all those things here. Okay, now the thing is that the lactose bacteria, the spores of lactose bacteria are present everywhere, and if you keep the milk open, sometime 3 to 4 hours, the lactose spores will fall there, lactose bacteria will become active and they will start the process, and more lactose will be taken in, glucose and galactose will be broken, glucose will be oxidized anaerobically, and the pyruvic acid will form, that will ultimately change into lactic acid, which all we are studied in class 11. And both lactic acid will cause the rise of lowering of the pH and the milk to convert into curd. Anyway, now the question is, that is the cell membrane has a little bit permeability for the lactose. So once one or two molecules of the lactose enters in the cell, they will directly come and attack the operator switch. Here it was our operator, here it was the polymerase for this, here it was inducer gene, and again here it was this, and Z, Y, here is the Lac Operon. The inducer gene will make a messenger RNA with the help of this. That messenger RNA is the messenger RNA for repressor. mRNA for repressor. R-E-P-R-E-S-S-O-R. Repressor. Repressor is a protein. Here repressor protein. Repressor protein will go and bind the operator. Here it comes and sits over. As long as repressor protein is here, these genes will not function. And no messenger RNA, polysystonic messenger RNA will be made, and no enzymes will be made, and no process will be functional. Now, the lactose molecule comes. Lactose diffuses into the cell. So as the lactose molecule diffuses, it comes and it will bind on the repressor. No sooner it binds the repressor molecule, the repressor is no more functional. The repressor is no more functional. What it is meant for, that function it will not be able to carry. That means the repressor will go out. The repressor plus protein. That is lactose, not protein, it is sugar. So this is called inducer. This lactose molecule is now known as inducer, which induces the repressor molecule into non-functional repressor. Okay. First, the switch was off. When the switch was off, when inhibitor protein gene made an RNA polymerase RNA molecule that is for repressor made protein. Repressor protein goes and binds the operator and the switch is off. Now the lactose molecule enters. As the lactose molecule enters, it goes to the repressor molecule and it changes some conformational changes into the repressor molecule and it will no more be functional, so it detaches from it and the operator becomes on. So now I am removing all these things. All these things are not required. So our operator is now active. As the operator becomes active, it will make the structural polycistronic mRNA. And the polycystonic mRNA has information for beta galactosidase, permease, and transacetylase. Galactosidase will break lactose into glucose plus galactose. Permease will increase the permeability and more lactose are entering. Permease increasing the permeability and leading more lactose to come in. Till when the process should go? As long as it is getting lactose molecule from outside and as long as it is having need of lactose molecule or glucose and galactose, the permeability will be maintained and the lactose molecule will go on entering into it and this will be broken into the glucose and galactose. Suppose the lactose molecules are over, there is no more lactose molecule left outside in the surrounding. The milk was only a drop. Now what will happen? The final lactose molecule is here. That lactose molecule will be brought here and that will also be broken. No sooner lactose is removed from the repressor, it will go and bind here and the process will again shut. Which may खुल गए थे ये and again now the switch will be turned off. Beautifully made system, that is wherever it was required it will be turned on and wherever its requirement is over it will be turned off. So this is how we come to know that how a particular gene becomes active and how a particular gene becomes inactive. These kind of genes which are normally off and become active are called negative operon. And there are some genes which are normally on and their function has to be turned off when functions are, they are called positive. Okay, so now there are so many different type of things we already discussed and there are so many different type of operons are studied so far. In our book there are 4 or 5 now examples are given. Lac Operon is there, tryptophan operon, tetraphan operon is written, then ARA that is ARA operon is there, histamine operator that is his operon, valine operator is are there. So many operons are given in our book and how the operon functions, these things we already done. So operon are actually a machine which is actually controlling the functioning of a particular gene. Now most of the cases in any cell the genes are shut off. They will be turned on only when a particular type of induction or information comes from outside. Okay, now since these cells are not meant for all those things, those type of informations are not reaching here, so these will no more be able to make HCl and pepsin and trypsin and lipase and what you call hydrochloric acid and many other type of things. So as we said, we got a function, our body has got capability of make so many different type of proteins, so many different type of functions are there. All these functions are regulated in our body. How they are regulated? There are 2 kinds of genes in our body. One, those which are always active. Those genes which are always active are called housekeeping genes. Like the genes responsible for digestion, the genes responsible for respiration, the genes responsible for muscle contraction, the genes responsible for like in plants it is for photosynthesis. These genes are all the time required. And there are some genes which are required as and when they have to be switched on and switched off. Okay, so this is how we have studied the various type of the processes. So if we study about the genetics, we are starting from the Mendel. Mendel said something about the factor, but he did not know where the factor was. Then later we came to know that the factors are actually located in the chromosome. So one solution is found. Later on we came to learn that the factors are actually the DNA, that is made up of DNA. So the second solution came. Then we came to know that one DNA has got so many different, the DNA molecule is very large, and then it has to make the function, it has to make the enzyme, that is the protein. So DNA has to make another type of, another species of the nucleic acid, that is called RNA, and the RNA will make function. And one DNA can make so many different type of RNA. So all those information we are step by step we learn. And now the last was the most important one, that how a function regulated. Why only a certain thing is functional in certain places and not on the other places. Now you see, if there is any deferment of any function, you see how bad the effect will be. And sometimes it happens. And that is why we are getting aging, we are getting some type of cancers, we are getting some type of defects, genetic defects and others. Okay, so that is all for today. Tomorrow's class we will study Human Genome Project. That is another very important topic. So you be there in tomorrow's class. Only 3 children.